This is the discussion of perhaps the most popular metabolic pathway of all time, and I think that's for a good reason, as I will discuss later on. The so-called Krebs cycle. It is actually interesting to note that the Krebs cycle can refer to two pathways because the guy named Krebs, the scientist Hans Krebs, discovered two cycles. However, since this one has a lot of implications, this turned out to be the popular Krebs cycle. Now, if you want a more specific term, you could also call this as the citric acid cycle, simply because there's citric acid or citrate in it, or the tricarboxylic acid cycle, which actually, in fact, refers also to the citric acid molecule. Notice this one, this is actually the structure of citric acid. And if you count the COOH groups in it, you find that there are three of them. So if you want to describe it based on the number of COOHs, you can call it the tricarboxylic acid. So essentially, this one is actually referring to citric acid as well. So first, for context, we should take note that the location of the Krebs cycle, or the citric acid, or the TCA cycle, is the mitochondrial matrix, so that's written here. And just to give you a quick view of what a mitochondrion looks like, I made here the simplest drawing I possibly could, which would give us the parts of the mitochondrion we need for future discussion. The outermost layer is called the outer membrane. That's easy, outermost layer. Then apparently that the way we call this outer membrane is because we have an inner membrane, which is this one. In between the two membranes here is a space, so it's literally this space right here. And interesting because we just call it as the intermembrane space, or literally the space between the two membranes. We could also abbreviate this as the IMS. And then the area to the inside of the inner membrane is called the matrix. So if I just said that the TCA cycle happens in the mitochondrial matrix, we should imagine all of this happening in the innermost region of a mitochondrion. Now, you have to know that the reason why we're discussing this is because this is part of cell respiration. However, this is not the first pathway that's related to cell respiration, as the first one I've ever discussed, if you're watching my videos in the arrangement they are listed in my playlist, is glycolysis. So in glycolysis, which is supposed to happen in the cytosol, we start with glucose and then we produce pyruvate molecule, actually two of them if we want to be accurate. And if you want to proceed with aerobic respiration, we want to send the pyruvate to the matrix or the mitochondrion, wherein the enzyme pyruvate dehydrogenase will convert pyruvate to acetyl-CoA. This has been discussed in the Fates of Pyruvate video, wherein as a byproduct, there's the release of carbon dioxide as well as the production of NADH. And since pyruvate has three carbons and then we remove one of those carbons, remember that acetyl-CoA at the moment has two carbons. Now, this is now the first actual step of the Krebs cycle, or the TCA cycle, wherein we imagine this acetyl-CoA merges or combines with oxaloacetate, which is a four-carbon compound, and by the power of the enzyme citrate synthase, they become a molecule of citrate. So, as the math here implies, the carbons of citrate is just the sum of these two, so 2 plus 4 is 6. So actually, if you go back to this molecule of citrate and you count the carbons, it should be the same. So 1, 2, 3, 4, 5, 6. So yeah, so far so good. And in the next step, citrate is converted by aconitase to isocitrate. Since, as the name implies, isocitrate is just an isomer, it should still have the same number of atoms, including the carbon. So this is also a 6-carbon compound. Now, if you have already a separate diagram of the Krebs cycle and you have in your picture or your figure two arrows here in between, that is not without impact or not without merit, because that's actually true. What actually happens here, why we get an isomer as a product, is that initially we remove water, so there's a dehydrogenation, sorry, dehydration, I mean, step, followed by the return of that water, or a hydration step. So you might think like, what did we do? We just wasted time. We removed water and then we returned it. But if you go back to your organic chemistry and like remember that not all carbons are the same in stability, unfortunate that in order to conserve time, I cannot discuss all of the details. Long story short, we get an isomer instead of the original citrate after the two steps here. Those two steps are facilitated by the same enzyme. So that's why in some figures of the Krebs cycle, this is just depicted as one arrow. Now, in the next step, we have isocitrate being converted by isocitrate dehydrogenase into alpha-ketoglutarate, which is a 5-carbon compound. As a tip, every time you have a compound with gluta in its name, like glutaric acid, glutamate, glutamine, it means it has 5 carbons. Of course, we now see that from 6, we have now 5 carbons. So if we ask, where did that 1 carbon go? It's actually released in the form of carbon dioxide. That is because isocitrate dehydrogenase also has a decarboxylase function. Actually, it's very similar to pyruvate dehydrogenase because even though the name of the enzyme is a dehydrogenase, it actually does decarboxylate as well. So, like, the way this works and this works is kind of parallel. Okay. Not only that, I did mention a while ago that pyruvate dehydrogenase makes NADH, and guess what? Isocitrate dehydrogenase also does that because the idea of redox is consistent supposedly across the board. So the idea here is that since dehydrogenase means isocitrate to alpha-ketoglutarate must have involved some oxidation, there must be an accompanying reduction, and this is not supposed to be new anymore to us. And so that reduction is what led to the conversion of NAD to NADH. Hopefully you find this convenient that whatever we discuss in the Fates of Pyruvate video is just like repeating here, just in a different pathway. Now, from alpha-ketoglutarate, we have the enzyme alpha-ketoglutarate dehydrogenase converting this to Succinyl-CoA, of course, meaning that we must add a coenzyme A for that to happen. And here, we once again have the enzyme dehydrogenase. And it would be helpful for you to know that Succinyl-CoA contains 4 carbons. So here, I'm actually trying to say that whatever isocitrate dehydrogenase did, alpha-ketoglutarate dehydrogenase will also do, such as 1. The removal of carbon dioxide, that's why you notice from 5 carbons we now have 4, and also the fact that this is an oxidation process. If this is oxidation, that means there must once again be reduction. Thus, you notice we produce NADH once again, just like this one. Let me encircle the NADH because we're going to count that later. Okay, so at the moment, we now have Succinyl-CoA. Now, you will actually notice we're now back to the original carbon count. A while ago, our starting point in this cycle was oxaloacetate. That's 4 carbons. Now, we're back to 4 carbons and expect it would be 4 carbons all the way to this one. So, this is actually the reason why you call this pathway as a cycle. When you say cycle, it should not end up with something larger than the original or smaller than the original. Okay? And now, of course, you may ask or you might leave a remark. It got larger along the way, right? From 4, it became 6. That is true, but that is just part of the process. It didn't end there. And then notice from 6, it shrank to 5, and it shrank back to 4. And now, what's the idea here? Since at the end of the day, the cycle is 4 carbons back to 4 carbons, what did we effectively do? What we actually did was basically feed this acetyl-CoA in the cycle and then kind of churn it out as carbon dioxide, such that if you think about it this way, what we just did is to convert the 2 carbons of acetyl-CoA to 2 molecules of carbon dioxide. Or, in other words, we have oxidized it to carbon dioxide. That's why in some textbooks they would refer to the Krebs cycle as the process that allows the complete oxidation of glucose. But why is it stated like that? Because come to think of it, I already know at this point that one of the purposes of the Krebs cycle is to convert the carbons of acetyl-CoA to carbon dioxide. But if you think about acetyl-CoA, where did this come from? From pyruvate. Where did this pyruvate come from? From glucose. So, in a way, basically, these carbons that you made into carbon dioxide or oxidized into carbon dioxide originally came from glucose. Thus, this definition is legitimate. Well, before we get too ahead of ourselves, let us not forget I have yet to discuss the rest of the pathway, so let's proceed. So from Succinyl-CoA, we will uh, convert it to succinate. That means the removal of the coenzyme A via the enzyme succinate-thiokinase, sometimes called Succinyl-CoA synthetase. Now, it so happens that alongside the cleavage of the coenzyme A, the energy that is generated from that allows the phosphorylation of GDP to become GTP, which is equivalent to one molecule of ATP since they are of the same rank. They are both nucleoside triphosphates. And if you remember my discussion in glycolysis, any step that allows the synthesis of a triphosphate, just like ATP, using a high energy substrate is called substrate-level phosphorylation. So this step right here is literally an SLP process. Next, we have succinate converted to fumarate via succinate dehydrogenase, and once again you have a dehydrogenase enzyme, meaning this must be oxidation, so there must be an accompanying reduction to that. In this case, instead of NAD, we use FAD, okay, and FAD is converted, of course, to the reduced form FADH. This is actually the only step in the Krebs cycle that produces FADH. Also, this is the only, let me go back to this a bit, this is the only substrate level phosphorylation in the Krebs cycle. Now, once we do produce fumarate, we have the fume, that's easy to remember. Fumarate is converted by fumarase into malate. It's not really that remarkable compared to the other steps. And then malate is finally converted to oxaloacetate by malate dehydrogenase, closing the cycle, and we go back to oxaloacetate. And since we have once again another dehydrogenase, there must be an accompanying production of NADH by reduction. So this is another one. So in Krebs cycle, it's actually easy to remember the enzymes here. Out of the 8 steps, there are 1, 2, 3. If you consider this as one step, okay? 1, 2, 3, 4, 5, 6, 7, 8. 4 of them have dehydrogenase enzymes. And doesn't it make sense that it's those dehydrogenase enzymes that produce our reduced cofactors? And thus, the logic is consistent all throughout. So, for example, you have an enzyme without dehydrogenase, aconitase. Do you see a NADH or FADH? None. Citrate synthase. Do you see a FADH or NADH? None. Fumarase. Do you see a FADH or NADH? None. So if you want to memorize this faster, or like make sure that you don't flip any steps up, as long as you have a dehydrogenase, remember you're for sure gonna generate a reduced cofactor. Okay, and again, just for this one, since this is the only SLP, always correlate the succinate-thiokinase enzyme to the generation of the GTP, such that in total we have 1, 2, 3 molecules of NADH, 1 molecule of FADH, and 1 molecule of GTP. This will be important once we compute the overall ATP yield for cell respiration in another video. Now, there is still something I would like to say about the Krebs cycle. As you see, some references refer to the Krebs cycle as an amphibolic pathway, meaning it could act both as a catabolic and as an anabolic pathway. But let me clarify that it is not the actual cycle that is anabolic, because if it were anabolic, your product should be greater than 4 carbons, which is not. If it were catabolic, your end product should be less than 4 carbons, which it is not. The idea of calling the Krebs cycle as amphibolic is that we think of the Krebs cycle as a bridge that connects several anabolic and catabolic processes. So, for example, you may have watched already my recording in gluconeogenesis, and I did mention that in order for pyruvate to go back to PEP, that is, to reverse step 10 of glycolysis, we must first synthesize oxaloacetate. So literally, we cross the Krebs cycle just for that in order to make GNG happen. And remember, gluconeogenesis is an anabolic process. Or for example, as I will teach in the future recordings for lipid metabolism, excessive amounts of citrate, whenever that happens, will lead to the synthesis of lipids, be it fatty acids um, as well as cholesterol. Or for example, okay, so this is, well, synthesis, so that is anabolic. Also, Succinyl-CoA, when we go to even more advanced discussions, is a starting material alongside glycine for the synthesis of heme and other so-called porphyrins. So since I just said synthesis, this is once again an anabolic pathway. But so far it's all anabolic. Another example is alpha-ketoglutarate being an assistant molecule to some amino acids, which will overall yield or produce the catabolic pathway for amino acids. So this is catabolism. So yeah, sometimes it's anabolism, sometimes catabolism, therefore justifying the term amphibolic. So think of the Krebs cycle as like, again, so maybe if we think of pathways as roads, you can think of this as one big crossroad or probably a rotunda where if you don't have this pathway, you will disconnect so many things. Okay. Also, sometimes we call the Krebs cycle as the central hub of metabolism, simply because most of our biomolecules that we could use for energy meet here. For example, we know that glucose could be used to synthesize uh, ATP. Okay. Uh, for example, we have pyruvate entering the mitochondrion, and then it will become acetyl-CoA. Then we enter the Krebs cycle. So carbohydrates can enter the Krebs cycle by this process. As I will teach in the future or in the next videos, fatty acids could actually be broken down into molecules of acetyl-CoA, thereby also entering the Krebs cycle. And actually most of our amino acids have the capability to also enter the Krebs cycle, therefore justifying the fact that our biomolecules can all meet here. That's why it's a central hub, like a meeting point for all of our biomolecules, because we do know that once we do produce these cofactors, they will ultimately lead us to the generation of energy. But don't think that this is the final pathway for cell respiration, because in order for us to truly convert these reduced factors to ATP, which is our end goal, we must first encounter the pathway called the electron transport chain. And that is what you will find next.